Hello, and welcome to a quick Chapter 4 slideshow lecture overview, where we're going to be talking about the things that go on in cells, prokaryotic, eukaryotic, plant, and animal. The chapter started off with some basic information about cell surface area to volume ratio, and we talked about this earlier on, I believe, last Friday's class. And the most important thing to note is that plant and animal cells tend to be quite a bit bigger than the prokaryotic cells that we see within nature. And then obviously they're all built of the macromolecules, proteins, lipids, carbohydrates that we talked about in Chapter 3. And we can use different types of microscopy work in terms of electron microscopes and light microscopes to see both cellular structures. We can see some nucleuses or nuclei in these different types of cells, but also we can start to see different types of organelles such as Golgi apparatus and cytoskeletons, and even specific aspects of cells like cilia when we take photos of these cells. So we need to know that prokaryotic cells are the basic fundamental structures of life that first colonized Earth a few billion years ago. And because they are both simple and very dynamic with responses to the environment, they're not that specialized. They don't have a nucleus. Their DNA is contained within the nucleoid. They really don't have any membrane-bound organelles, so their metabolism and their characteristics aren't very complex. The cytoplasm is obviously bound by the plasma membrane. Other things to note is the flagella / the cilia that you often see on prokaryotic and eukaryotic cells are meant for both motility and surface area. The ribosomes are obviously going to be useful for protein production, and the fimbria are different ways of bacteria being able to communicate with each other. Now, eukaryotic cells are far more complex because they not only have a more specialized metabolism in terms of energy production and protein synthesis, but they also make up specialized tissues and organs and organisms. So they tend to have a lot more diversity within their structure and their function and their genetic expression. And they tend to have many membrane-bound organelles as a result. Some of the organelles that I think are important to touch upon that we might not talk about much in this chapter, one would absolutely be the centrosome, which are microtubule organizing centers. These will be very helpful to consider when we get into cell division. Lysosomes and peroxisomes are two different vacuoles that we find in plant and animal cells that are useful for not only digesting dead or recyclable cell matter, but also it's a good way for some cells to use for immune systems and defenses. So peroxisomes and lysosomes are very useful in dendritic white cells or macrophages that actually go around your body and eat bacteria when you are sick. And obviously we can talk about the cytoskeleton later. Now, within plant cells, we have a lot of the similar properties, some differences. We have a central vacuole, which is going to be very useful for storing energy, but also water, because it's very important that plant cells stay hydrated for photosynthesis. We also have the chloroplast within plant cells for photosynthesis. We have plasmodesmata in the cell wall, and that mentions obviously that plants have cell walls made up of cellulose, polysaccharide, which we learned about in Chapter 3, and that helps with rigidity. Obviously, plants need to be both flexible, but pretty sturdy and rigid in order to grow upwards for photosynthetic output and primary productivity. So the plasma membrane that we see in all three cells is selective in terms of what is able to enter and exit, and it allows also the passage of things that cells absolutely need, like oxygen and nutrients and waste, to make sure that cells can be responsive to their environment, but also maintain homeostasis, or a pretty constant internal environment. The general structure is a double layer of phospholipids. We talked about phospholipids briefly in Chapter 3, but generally they create a hydrophobic region and a hydrophilic or water-based organism that helps regulate this. And we can see within the structure that we do have gates, or we could call these integral proteins or channel proteins that allow things to come in and out. And generally we have a lot of receivers or receptors within the cell membrane that allow us to sort of communicate with other cells, receive stimuli, and also allow the cell to respond to environments. As cells get bigger, their surface area to volume ratio decreases, which means that it's harder for cells to respond to those environments because the metabolism or the volume of the cell increases, but the surface area is not able to keep up at the same rate. So cells tend to stay small and form tissues out of many of them rather than have large cells. Now, when we look at eukaryotic cells, especially eukaryotic animal cells, we can really talk about the endomembrane system and how protein expression occurs. Within the nucleus, we have our DNA, which is wound around, there we go, different protein complexes called histones. And these histones use a chemical charge opposite of DNA to attract them, to wind them around. And then these histones, based on hydrogen bond interactions, coalesce together to help condense and organize the DNA. The DNA in your cell is miles of genetic information long, and we need to fit all of that into a very small package. So we use chromatin as a way to sort of condense and coalesce and organize all of that genetic information. I believe I made an analogy to Harry Potter, how we take 7 years of Hogwarts and divide it into 7 books. And the inner and outer membrane of the nuclear envelope helps protect all of this genetic information from damage, and it also makes sure that we regulate genetic expression well. Now, ribosomes are going to be integral to being able to produce proteins. Ribosomes are made in the nucleus, and they leave and go out into either the cytoplasm or embed themselves in the endoplasmic reticulum, specifically on the rough ER, and they are going to be making protein synthesis as a result. We have a large and subunit of proteins that come together to form ribosomes, and between the large and small subunit is going to be where mRNA, the transcript of DNA, is read to create a protein. Now, in terms of the endomembrane system, which is really fundamental for protein production and regulation, and also allowing metabolic processes to occur, there are a couple steps. We have the nuclear envelope, which regulates protein production and things going in and out of the nucleus to make proteins. We have the endoplasmic reticulum, where proteins are made, the Golgi apparatus that packages and modifies proteins, lysosomes, which are used for helping break down proteins that might not be correctly produced, or proteins that have gone beyond their shelf life, vacuoles for storage, and also the plasma membrane, where proteins can be embedded or released into the extracellular space, maybe another cell, a tissue, the bloodstream. All of these components are connected through transport vesicles, which are basically tiny little bubbles of the plasma membrane that allow fusion between these different types of organelles, but also packaging to separate and protect the protein product. The rough ER accounts for most of the total membrane in most eukaryotic cells, so it's very embedded, it's very folded, and there's two types that we find outside of the nuclear envelope. We have smooth ER, which lacks ribosomes, and we have rough ER, which is embedded with all of the different ribosomes, and they do different things based on the cell's needs and also what type of cell we're looking at. So here's a good example of smooth ER, here's a good example of rough ER, and you can see closely that we have all of these ribosomes, which are embedded sort of on the external layer of the cisterna, which is a fancy word for the membrane, And as we make our proteins, those proteins are going to be created into sort of the lumen or the inner folds of the membrane. And then as the proteins are ready to be transported and packaged, little bubbles of the membrane will fuse out, forming a transport vesicle. The smoothie R we find in a lot of cells like liver cells and kidney cells, and they basically synthesize lipids, but also they're really important for detoxifying drugs and poisons within the bloodstream or within cells. It's also very common to find in muscle cells because the smoothie R stores calcium ions, which are released and used to make the actin strands in your muscle fibers move and flex. The rough ER obviously has ribosomes, which are going to be creating the proteins, and they're going to then allow transport vesicles to surround those proteins and go off to different parts of the cell. Basically, this is the factory of proteins for the cells. We will sometimes make glycoproteins and not just proteins in the rough ER because the carbohydrates that we might add to the proteins, either in the rough ER or the Golgi apparatus, can be used as the messaging and receiving aspects of the cell membrane. So these carbohydrates add more specificity to the shape and structure of those proteins. We then send the proteins and the transport vesicles to the Golgi apparatus. The Golgi apparatus modifies the protein in terms of adding carbohydrates, cleaving or rearranging amino acid strands. They can manufacture other types of macromolecules, and then it really sorts and packages those materials when they're finished to their final destinations via more transport vesicles. You're going to often see transport vesicles coming into the Golgi apparatus, which we call the cis side, or the receiving side of the Golgi apparatus, and they will fuse into, again, the cisterna, or the plasma membrane of the Golgi apparatus, and release the packages or the proteins or glycoproteins into the Golgi apparatus. Each of these folds could be alluded to or made as an analogy of just protein packaging on an assembly line, where each of these folds is going to be doing some aspect of change to each protein using enzymes and metabolic processes. When the protein is finally packaged and finished, it's going to be shipped via a transport vesicle out of the trans side or the shipping side. Now, proteins die, proteins are made incorrectly, there might be waste accumulating in a cell. We use lysosomes as a way to break down macromolecules and also break down foods. So lysosomes can hydrolyze proteins and fats and polysaccharides and nucleic acids. A lot of different things that we get from our food and or from our bloodstream that we could either use to rebuild aspects of the cell or to make sure that incorrectly made things are not put into the cell, compromising their form and function. Lysosome enzymes within that small membranous sac are very acidic. So they have a very specific niche in terms of their form and function. The enzymes will work in that acidic environment, which is separate from the non-acidic environment of the cytoplasm. Some types of cells can engulf other cells via phagocytosis, and this is often seen within macrophages, which are a type of white blood cell that go around and basically eat bacteria. And when that happens, a lysosome might fuse with those vacuoles or those cells, and they can digest it. And this allows not only an immune response, but also, again, the lysosomes can use enzymes to recycle that cell's own organelles and macromolecules to then create new organelles and macromolecules. We call this autophagy, especially if the cell is doing it to its own organelles. So really, we're creating a self-sufficient system within each cell. And we can see this here where a lysosome uses phagocytosis at the external cellular membrane. It pinches off, forming a food vacuole. The lysosome with digestive enzymes can then fuse with the food vacuole because they're both of that phospholipid bilayer. And then the digestion occurs of those macromolecules. And this can happen at a cellular level. We're going to engulf some bacteria as a dendritic white cell, or it could be things as simple as absorbing some macromolecules in your bloodstream. And the same thing can occur with maybe a mitochondria that stopped working or needs to be replaced, where we can have the lysosome fuse with the mitochondria and break that organelle back down into building blocks to be reused and recycled. There are other types of vacuoles that we find in organisms. So contractual vacuoles are found in many freshwater protists or bacteria that pump out excessive water due to osmolarity changes. So water is constantly rushing into these organisms because they have a higher solute concentration than fresh water. And also central vacuoles that we find in many plant cells hold organic compounds in water that allow the plants to always have a storage of energy and water for photosynthesis or cellular respiration. And these take up a huge part of, obviously, the plant cell because they need so much water for photosynthesis. You should feel really comfortable with thinking about how DNA is expressed into mRNA that leaves a nuclear pore, enters either a free or bound ribosome, which is going to create a protein that either stays in the cytoplasm or is going to leave or be embedded in the membrane. A transport vesicle is going to fuse to the cis side of the Golgi apparatus, which will modify and package that protein, leaving as another transport vesicle that will go to the plasma membrane. We have two types of organelles found in eukaryotic cells that are important for energy, but also the carbon cycle at a cellular and global level. So mitochondria are going to be the sites of exergonic reactions that we call cellular respiration, where we use oxygen to break down, oxidize, reduce glucose to generate ATP, which is the cellular currency of cells. Oppositely, chloroplasts, which we find in plants and algae, are where we're going to be endogonically breaking or building up CO2 into glucose using ATP. These two processes are mirrored and opposite, and we'll talk a lot about them in chapters 7 and 8. The mitochondria and chloroplasts are interesting in that there is some evidence that these used to be independent prokaryotic organisms that formed a symbiotic relationship, hello chapter 41, with other cells. Here's why. Each has a double membrane, which seems or appears to be a vestigial remnant of when they were their own cells. They both have free ribosomes within their organelles, so they can produce their own proteins. They both have circular DNA molecules, which is like bacterial cells, if we go back and look at the beginning of this slideshow. And they can grow and reproduce independently without considering the roles of mitosis. So they can grow and reproduce without really needing the eukaryotic cell host to allow them to do that. The endosymbiotics theory is the idea that at a very early eukaryotic cell engulfed a non-photosynthetic prokaryotic cell, and that formed a relationship, an endosymbiotic symbiosis within that host. Endo means within, symbiont means that they benefit each other. The host cell obviously gave the mitochondria, the non-photosynthetic prokaryotic cell, safe space, and the eukaryotic cell got energy from the mitochondria. At least one of these cells, there's evidence that this eukaryotic cell happened multiple times, took up a photosynthetic prokaryote, probably some early ancestor of an algae, and that became the precursor of plant cells And we could say that that photosynthetic prokaryote eventually became what we see as a chloroplast. And this is how we think it probably happened. So there was a ancestor of a eukaryotic cell. The nucleus had already started to become a thing. We had the endoplasmic reticulum for protein synthesis, but we're lacking a way of obtaining energy. The non-photosynthetic prokaryote embedded itself within that cell. We created a symbiotic relationship where energy was exchanged for shelter. And then one of these cells obtained a chloroplast, which gave us both cellular respiration and photosynthetic capabilities. The mitochondria has, again, ribosomes, a double-folded membrane, and they also have DNA, which can be very useful for analyzing ancestry because mitochondrial DNA is maternally inherited. The chloroplast also has a double membrane with stacks of thylakoids, which we call granum, and this is where we use different types of proteins to absorb light and fixate carbon into glucose. Peroxisomes are another type of organelle that produce hydrogen peroxide and convert it to water, and this is very important for a variety of reactions within cells in terms of making metabolic processes more efficient. The last thing we have to talk about is the cytoskeleton. Cells move, cells are dynamic, their shape, their surface area, their volume change. They also have to rearrange their organelles for cellular division. They need to make and move proteins from one organelle to the next. The life of a cell is not static. It is a very dynamic. We have motor proteins that can walk along microtubule cytoskeletons, and these motor proteins use ATP energy to move vesicles between different organelles. These microtubules can be arranged to create new roads or avenues for vesicles to move from one organelle to another. We also have microfilaments and intermediate filaments that also exist within the cell. The microfilaments are the thinnest, and they are just two actin proteins that are together, and they're really important for muscle contraction, changes in cell shape. They are often analogous to the thin metal strands that you put in a camping tent. And they can be useful for the division of animal cells because they can help pull apart different organelles. Obviously, microtubules we talked about use for the movement of motor proteins, but they're also really important in dividing chromosomes during mitosis. And then intermediate filaments are the in-between fiber, and they're really important for anchoring things like the nucleus, maintaining the cell's shape, and sort of keeping the cell structurally intact. These do not break apart as easily. They're very fibrous. It's almost like steel rods, while these two are more flexible. The centrosome, as I alluded to, is the location near the nucleus that organizes microtubules, and that becomes very important during mitosis. Cilia and flagella are useful for movement. Flagella are very common, especially in eukaryotic cells, for movement, and cilia are very numerous and often increase the surface area of a cell, so they might be common in things like a small intestinal organ for increasing the absorption of nutrients. The cell wall, obviously, we talked about earlier, is super dense and structurally sound as a result of it being primarily built up of cellulose, but also some proteins, and that really helps the plant maintain rigidity, its shape, and especially keep the tissues intact when there is a deficit or excessive amount of water. And we can see how they sort of keep the cells intact and form tissues. This is why you can have a tree that's 200 feet tall that can both flex in the wind but also stays intact. There is an extracellular matrix of different types of proteins that connect cells to each other. These are things like collagen and fibronectin, which can be useful for things like clotting, but also keeping muscles intact and embedded and also attached to bones. And it basically creates this fibrous network of allowing cells to connect to each other. Plasmodesmata are found in cell walls, and they allow plant cells to communicate with each other. And within animal cells, we have three types of intercellular junctions that allow animal cells to work properly. Tight junctions are sort of like sewing machines that make cells very compact and tight next to each other. So if this is the small intestine, we don't want intestinal fluids leaking into the extracellular matrix. Gap junctions allow for quick, easy movement of things from one cell to the other, and desmosomes are useful for cellular communication. I hope this has been a chapter that has made sense in terms of how the structure and function of cells work. Let me know if you have any questions about any of this material.